Hi, everyone. My name is Brook Mangano, and this summer I work at the MagLab at UF under my mentor, Dr. Joanna Long, and alongside postdoc Dr. Maria Luisa Caldas-Figuera. I spent my summer studying lipid dynamics of pulmonary surfactant due to surfactant protein B. So we used deuterium NMR to understand how FTB induces an unusual lipid movement in pulmonary surfactant. Why use time and energy to study pulmonary surfactant? So pulmonary surfactant is necessary for lung function, and this is all because of surfactant protein B. It is the most vital component of the mostly lipid surfactant. So it's 90% lipid and 10% protein, and FTB is the most vital component because it induces this behavior. So from the lung cells, the pulmonary surfactant is released in lamellar bodies, and FTB allows it to unfold at the air-water interface, reducing surface tension. If there is too high surface tension, then our lungs will collapse. So as you can see here, this is the NMR spectra of samples with the protein and without the protein. You can see the spike right here, and that is what is called an isotropic phase. Without the protein, you don't see that. And this is what the isotropic behavior looks like. So method. We performed NMR on mixtures of deuterated lipids with synthetic surfactant protein B. So I made these mixtures and we needed them to be deuterated to see signal. So we varied pH between samples because we wanted to test if pH had an effect on the behavior. So as you can see here, the pH of the cells are higher, and then the pH closer to the air is lower. So if we wanted to see if this had an effect on the behavior. We also tested different temperatures to see if it was temperature dependent as well. And then most of my summer actually was spent trying to express and purify the protein from E. coli like the last presentation. So we used a vector that had the DNA that coded for the protein, and we had to try to isolate it from all the other proteins that it made. Results. Unfortunately, I didn't obtain enough peptides from the E. coli to be sufficient for NMR samples. We did get some, but this is not unexpected because the protein is very hydrophobic and it has unstable disulfide bridges. But we are working on trying to make that protocol better to have a higher yield. However, I did run NMR on the samples with synthetic peptides, and I ran 5 different pHs and then a control with no peptide. And as you can see here, all the different pHs look very different. So at a pH of 7, you see the most isotropic spike right here. Also, you can see that this is 35 degrees and this is 30 degrees, and they also look very different, but this is the same pH. So we also found that there is a very high temperature dependence as well. Conclusion, we now know that pH and temperature has an effect on the mechanism. At body temperature, the lipids were most isotropic going or at a pH of 7. And this is interesting because it is kind of the transition between the cell and the water. We still need to know what is making it act this way. So if we develop it from E. coli, it will be possible to put isotopes on individual amino acids to test if those amino acids are responsible. If we can do this, we can find other proteins like this and better understand lipid movement due to proteins. And if we also get it from E. coli, it will be safe to use for medicine instead of using it from animals, which is very hard on the immune system. I would like to thank you for your attention and my mentors, Dr. Joanna Long and Dr. Luisa Caldas-Figuera, and also Dr. Corona Johnson and the MagLab for making this a worthwhile, productive, and enjoyable experience. Thank you.